My name's Kate. I'm from little old Auckland, New Zealand, at Unitec. So I was quite thrilled to be. Thank you very much for inviting me to come along to the seminar and share some of the work that I've been doing. So what I'm going to focus on today is zoonotic hookworm in the free-roaming dogs from Tonga and the evidence that we have found for Ancylostoma ceylanicum. So just a quick little talk about Tonga, which is our main study location. So the main work done for this study is on the main island of Tongatapu in the Kingdom of Tonga. There are many other island groups, but Tongatapu has the majority of the population. In Tonga, the dogs are pretty much free roaming, often referred to as community or village dogs. So there's a few reasons for this. Some part is due to limited resources in terms of fencing on properties. It's also a cultural aspect where dogs are to be free roaming, and they tend to also do a lot of food scavenging due to a lack of commercial dog food products available in Tonga. So obviously there's some animal welfare issues involved here in terms of the preventative healthcare. So there is no permanent vet in Tonga available for animals' day-to-day care, obviously including for their agricultural stock. So there is the South Pacific Animal Welfare Group, which do pop-up vet clinics 5 times a year on Tongatapu, and there's a couple of vet clinic pop-ups that go to the outer islands of Vavau and Hapai. These clinics are very much focused on desexing to help with population control and management for the dogs and cats, and is one of the few opportunities that the local communities are able to get access to preventative healthcare for fleas and worms and that type of thing, and any other potential medications. Outside of that, the only support they have is from the Ministry of Agricultural staff, who are very much focused in agricultural animals and not in companion animals, but they are certainly doing their best to upskill where they can. So Tonga is again another very typical tropical climate, warm weather and wet seasons. And this type of thing is a pretty common outlook, especially when you go there anytime from October to April. Uh, Pretty normal life to be walking around ankle deep in water this type of season, which obviously is potentially a breeding ground for all sorts of zoonotic pathogens and those types of things. So I'm not going to go too much here because this has been pretty well covered by Richard, but probably the main things I wanted to point out here is with the canine hookworms, there is very high impact on the animals, quite severe levels of anemia. Morbidity is more common than not in young puppies with high burdens due to the level of anemia, which is obviously going to be a combination of the parasites and nutritional lacking as well. So Ancylostoma ceylanicum is our top is my chosen one for today, which we're going to focus on here. And as Richard and Juan have been talking about, there's not that much in terms of what we know about this parasite, or it's starting to become more well known, and it's obviously something that we need to build on. So, originally in 2019, a colleague and I started what I will refer to as a small pilot study in Tonga because there has been no work done of this of the style there in Tonga in terms of the animals. So, our main investigation here was actually not focused so much on the parasites, it was more trying to get some basic understanding of general health through biochemistry, PCVs, and the fecal floats were actually no idea what we're going to find. So our sample size was a little bit small in terms of, so we had 78 fecal samples, which we did the flotations on. And as I kept going through the samples, I just kept obviously finding hookworm positive after hookworm positive, and more than 70% of those samples tested did have hookworm eggs in them. Obviously, morphology wise, I can't tell that, well, definitely I can't tell the difference in terms of the eggs, and I believe it is quite difficult morphology wise there. The majority of these dogs were less than a year old, but that's with the infections. That's likely a bit of a bias here though, because it is a desexing clinic, so it is generally going to be the younger animals that come to that clinic. Uh, Nearly 90% of the dogs had some level of anemia or borderline, and this is potentially too combination of two factors here, one being the endoparasite burdens, but also the issue with nutritional accessibility for these dogs as well. So when we found this high level of hookworms, we were definitely interested, and that's where we developed to the stage two, thanks to somebody called Dr. Richard Bradbury reaching out to me asking if I wanted to do it again. Of course, I said yes because I love this type of thing. So the next step, so the idea was to take that step further and really look much more at the prevalence of the species, sorry, the prevalence of hookworm in general, what species were actually present in Tonga, and what sort of spread there was across the island, because our initial study was very much in one location. And obviously, the outcome of that is to look at the general population health for people. So, how is this affecting the Tongan community? What sort of transmission is potentially going to be occurring? So, this was a week-long study during October, during the wet season, and this was run through what we would call village clinics. So basically, we were at a different village on Tongatapu every single day, Monday to Friday, and then the following week, part of the team were able to go north to Vivau and get some sampling at a clinic there. All sampling was done while patients were under anaesthesia, and that was part of our ethical approval. Everything needed to be sedated or under anaesthesia. We managed to get a lot more samples here. So we had nearly 200 fecal samples to work with, which was great. And once we had these fecal samples, we separated them into formalin and ethanol so that we could transfer them or export them out of Tonga to Australia to James Cook University to do some higher level diagnostics on them. The blood samples, we did PCVs on the island so that we could have this to look at later with along our fecal samples. And then other parts of the blood were separated and sent back to Australia as well for some other testing. So the main two things we did here, well, the two things we did here for our fecal samples was we've carried out formalin acetate sedimentation to do our microscopy identification. And then we've also done our qPCR tests in order to look at what hookworm species are present there and a few other potential zoonotic pathogens or parasites as well. Very prelim results. We've just finished running the qPCR, so I just wanted to give you an idea of where we're at the moment. So out of those nearly 200 dogs, 80% were positive for Ancyclostoma caninium and 25% for ceylanicum. 18% had both of those parasites, or testing positive for both of those parasites. Brazilianese and Uncinerea showed up negative, and then we had our other pathogens that we tested for as well. So just to give you a little idea about the geographical distribution, you will obviously notice that Vivau has a much larger sample size. That is due to the fact that it was sampling over 4 days, whereas our other clinics were 1 day only. So of course the sample sizes were a bit smaller. Geographical distribution, this is quite important, not just for where these parasites are, but also considering accessibility to people for preventative treatment, whether it be for their pets or for themselves. So I'm going to focus on the 5 Tongatapu villages that we tested. And I did try to find out some populations of each of these villages for you to give you an idea of the size of them. It's incredibly difficult to find accurate information on here. So generally speaking, 500 to 2,000 people might be in the various different villages. Every single village has some level of agricultural component to it, whether it be cows, pigs, chickens. Very minimal in terms of sheep are available in Tonga, generally due to the climate and compatible breeds. So our first wee village up here is Kolowai. It is on the very western peninsula. I'm being generous by saying a 45-minute drive to Nuku'alofa. So Nuku'alofa is there in the centre, and that is probably the main source for resources, that type of thing. You do have little shops along the way, but if you're looking for specialist things, like generally pharmacies are really only found in Nuku'alofa. There's only 2 or 3 in Nuku'alofa itself and nothing outside of town. There's no pet shops, there's no vet clinic, that type of thing. So that's how far you're tending to have to travel. And then the next village we visited was Nuku Nuku. So these are both coastal villages, and it's probably worth mentioning that these two villages were quite severely affected by the 2022 Tonga eruption, which resulted in the tsunami. They probably suffered quite a lot of water damage here, and these samples were taken after that eruption occurred. Uh, Tokomalolo is our normal location, which is where our Ministry of Agriculture department is. So this is a very normal, sorry, not normal, this is very, very central and probably the closest to town. It wasn't surprising to see a lower percentage for our caninium there because there is some accessibility to preventative healthcare for dogs from the centre when they have products available for the public all the way down to Fuamotu, which is, if you look closely, you can see the airport strip there as well. So this is one of the most southern, well, probably the furthest you can be from town other than the East Coast. Um, Again, they are very, very far from resources. There's not a lot around there. The just something to note here in terms of the ceylanicum percentage there, that is this was a very, very small sample size. Unfortunately, only we had 15 animals for surgery or 15 dogs for surgery that day. So just to keep that in mind when you're looking at those percentages. And then the last one on here is Kolunga, which is on the very east coast. Again, at least 15 minutes drive from town. This is a predominantly a fishing village. They're a little bit more exposed to the ocean, that type of thing. And yeah, again, quite a far trip from town. So limitations. Sample size. So while we have definitely increased our sample size from our first initial pilot study, it is now that we've spread it over the geographical different villages, it does sort of make for smaller sample sizes. I'd love to be able to collect more for each of these villages and get a better idea there. However, it is very dependent on the situation and whether animals are actually submitted for surgery or not. I was really thrilled that we were able to expand the geographical coverage and go and do these village visits rather than just focus on one central location. Our original diagnostics were pretty limited and we were only able to do what we could do on island. So being able to export these samples to a more appropriate facility and probably a more controlled environment was extremely helpful in terms of being able to do our microscopy and our molecular work. So the main point, so obviously we've got a little bit more in depth to go with this data, but I think it's pretty clear that there is hookworm throughout Tongatapu from these prelim results. Acaninium is clearly the winner when it comes to prevalence, and it's found in all the villages. The proportion of ceylanicum is pretty high. I would consider a quarter of a sample population for ceylanicum pretty up there, and that obviously does cause a concern. There is very little work published on canine hookworms parasites within the Pacific region, so it's really hard to draw any comparisons based on the numbers we found. But obviously, the more work we all do in this area, the more we're going to have to compare to. I just wanted to mention in terms of moving forward, dogs acting as a reservoir host, this is potentially an issue that is going to be very, very hard to correct in well, not to correct, but to manage in a One Health perspective. The way the dogs are living at the moment and the accessibility to treatment and nutrition, that sort of thing, is something that's going to take a long time to progress, especially based on funding available to the country and that type of thing. The zoonotic risk is is very much unknown for people in Tonga because there have presently been no studies performed on endoparasites within the Tongan community. This is definitely something that needs to happen, especially now that we've pinpointed these particular hookworm species that are present there. Future work, I love it. Let's do more research. I'd really love that we could continue to do longitudinal monitoring on the situation in Tonga, especially in the dogs, as resources become hopefully more available and education and husbandry sort of things come into play. Environmental sampling, I think, is something that is worth looking at. There's certainly been some interesting studies from Brazil who did some work effectively testing the sand on the beaches and finding quite a number of hookworm even just within the sand on the local beaches. Obviously, we need to look at how these hookworms are and where they are presenting in people to see how what's affecting in what's affecting the local community as well. Some massive acknowledgements. There's been a whole lot of organisations involved in this work to date. I definitely want to do a special mention to my colleagues at JCU because we definitely wouldn't have made it to this next step without the help and support from everyone there. Cool. And that is me.